Thank you very much. Let me check it out whether Professor Julius is here. Yes, I found him.
I'm here.
Yes, thank you very much. So it's a great honor to find here our last keynote speaker from industry of the research organization, Dr. M. Julius Hossain, research scientist from European Molecular Biology Laboratory, Heidelberg, Germany. We have also amongst us Professor Dr. Muhammad Mamun Rashid from University of Dhaka for sharing this session. Dear audience and the participants, may I now request Professor Dr. Mamun Rashid to introduce Professor Julius Hossain and continue the session.
Thank you, Professor Jack. It's really interesting that today I'm chairing a session with whom actually we started our student career in the same organization and same institute. And also we have pursued our PhD together. So I personally know Dr. Julius for quite long period of time, and maybe we plan to meet several times virtually, and today it's an opportunity for us to meet virtually. And how to introduce him? Actually, he is one of our genius in CSCDU, Dhaka University Computer Science and Engineering Department. Although now he belongs to different organization, but he is a genius. He proved his research and other things in many different areas. And right at this moment, Dr. Julius is sitting as a scientist in the European Molecular Biology Lab. There is a research scientist over there, and as I said earlier, we together pursued our PhD in Kean University, and after that he joined Dublin City University as a postdoctoral fellow. Till now, Dr. Julius has published a number of papers in Nature, Science, and some of their impact factor is more than 42, 41, which is really encouraging for us. And today, Dr. Julius is going to tell us about the role of bioimage informatics to study fundamental biological questions. I hope you will enjoy this session, and hopefully we will have you will have a lot of questions with him also. So may I request our last keynote speaker, to deliver it. Thank you.
Thanks to my friend, Dr. Mamun, for his generous introduction. And I would also like to thank the organizer for inviting me for this talk. So if you stop sharing, I will start sharing my screen. Yeah. So my screen is there.
Yes.
Yeah, yes, it's shared.
Let me separate it to. Is it in presentation mode? No.
Yeah, it is.
Oh, okay. So somehow I'm having, let me do it again.
I think we have some technical difficulties. Let's wait for a while.
Yeah.
Yeah, okay. Now, আমাদের এখানকার connection unstable হয়ে
So somehow I cannot see that it's going to be in presentation mode. Is it the same there?
It's not in the presentation mode.
Yeah, actually earlier it was in the presentation mode. Julius,
Just
maybe
try.
Yeah.
Trying one more time. Okay, let me use my, yeah. Yes. Maybe I first go to presentation mode and then try to share.
I think you go first in presentation mode with the PPT slide and then you start sharing.
Yeah, that I will do now. Yes.
Yeah, it's okay. You need to share your screen.
Yes. Is it the right one? You see?
Yes, the PPT title is fine, but it's still not in full screen mode.
Okay. Let me stop my, sorry for the jet, I don't know what. So I stop. Yes.
Dr. Julius, I found there
Yes.
a button, resume slideshow, while you shared. Maybe if you click there, then it will work, I believe.
Do you see it now?
It's perfect. Perfect. Please
Okay.
go ahead.
Okay. I had to swap my screen. So my laptop is now my main screen. Yeah. So sorry for the delay. I have to check my time as well. So in this talk, actually, I would like to introduce the bioinformatics, bioimage informatics, and the role of it in studying fundamental biological questions. So yeah, so I start with a press release in 2008 after we published a manuscript in Science. So the highlight was that EMBL scientists show that. Hello, can you?
কিরে পান্ডার দল তুই কি এতে নাকি জুমে নাকি এই যে বায়োইনফরমেটিক্স
Hello.
আমি মাত্র ঢুকলাম
Please.
তুমি তো বই আর 10,000 এর লিংক আমি তো জানি
I think
Please mute your mic.
I think কেউ একজন কথা বলতেছে, একটু মিউট করতে হবে।
Yes. So
Somebody else is talking.
so the main point here was that so the highlight was that EMBL scientists show that mammalian life begins differently than we thought. So mammalian life starts when maternal and paternal genome connect together. So what we have seen there, that after fertilization or conception, so there are two genomes. One is coming from mom, and the second part is coming from dad. So these are two pronuclei, and then they fuse together, but still, especially when they go to two cell, from zygote to two cell, this maternal, which is red, and blue is paternal, they are still separated. So with that, we could change our knowledge that in second cell division, still maternal and paternal genome are separated. So what is the impact of this research? So we could redefine the beginning of life. That means that people used to believe that just after conception or in zygote, maternal and paternal genome start mixing and fuse together. So that's the starting of life. And in many countries, the definition was like that, but we proved that it is not the case, and it is not fully accurate. So then that also impact the potential legislation, because this definition of life define how long you can work with human embryo. So in many countries, most of the countries in the world, government doesn't allow to do research on human embryo, because they think that this is the beginning of life. So life is already there. And now, because we published this with mouse embryo, feminists asked us to prove it in human embryo, so that they could also ask government to change the legislation, so that some sort of work, research work, with in vitro fertilization and also human embryo can go ahead. And this also impact the biology textbook. So what I want to make here, that the fundamental biological question is something that impact a lot in the field. So to describe those topic, I have selected three different examples. So one is in organism level, where we work with embryo or full organism. And a part of embryo is a cell, so where you have a lot of subcellular components. So this is a small part, but cell is the most complex machine in the world, and it has all the information that can control human life and the behavior of human life. And then we can also, I will also give an example of subcellular compartment, which is we call protein complexes, which is very tiny structure inside a cell. And the reason of doing this is that their size is different. So what you can see and the way you can see should be also different. So this is an example that if you are interested to do research on a different field, then you need to have the capacity to look at that. So we look at things which is 1 nanometer to several micrometer or millimeter range. And you cannot do it with any microscope. So to look at something which is very small, you need to go beyond existing technique. And that's the reason there were different microscopy techniques established. So at EMBL, actually, we also contributed development of cryomicroscopy and also cryo-electron microscopy and super resolution microscopy. And two of our former alumni got Nobel Prize for that. So first, I would like to talk about the starting of mammalian life, which is actually you have fertilization and then there will be several cell division and organ formation and complete baby formation. So what happens here that you start with a oocyte that comes from mom and it goes through a couple of non-uniform divisions. So they had to have exact different number of chromosome inside the egg. And then the fertilization happens. So after fertilization, you will have one from maternal, the other one from the paternal, and they try to come close together, and then it divides into 2, 4, and do some sort of organ formation and later stages. So this is the process of starting of life. So one important limitation that we have noticed that in case of mammalian, even human, so only 30% of the conception goes to successful birth. So there are 70% loss in different stages of embryonic development. So the main reason for this is aneuploidy. That means the chromosome are not divided uniformly. So they are kind of heterogeneous. They call chromosomal defect. So to look at those things, you need a powerful microscope and methodology to observe the early stages of embryo so that you can understand what's the reason of those errors. So with this, we tried with the existing microscope. So, but in our group, we had to develop our own microscope, which is light sheet microscopy. And with that, we could look at the progression of human embryo in early stages for a couple of weeks. And that allow us to find what are the reason for error. So we try to track individual chromosome through centromeres. So if you see, one is coming from mother and the other set of chromosome coming from father. So by tracking, we could find out that even after separation, they stay in the separate compartment. So they don't mix together. Okay, so this green and magenta do not mix together. So that was the point of discovery. So we need to do a lot of computation, but this is not the topic I will discuss a lot, but just roughly that if you look at that maternal and paternal centromere, they don't mix together much, but we need to have quantification. And in different stages of embryonic development, zygote, 2-cell, 4-cell stages, we quantified and we found that in 2-cell stages, the mixing is not a lot. So they largely remain separated. And this we also did using different experiment where we marked only one centromere, and then we try to fit a plane so that we can separate. And we also looked at that they don't mix together in early stages, and we compare it with the simulated results. So with that, we concluded that the maternal and paternal genome remain separated, but we also try to understand why they remain separated. So people used to believe that there is a machine which called spindle. So in early stages of human development, there will be only one spindle, but we figured out that it's not 1 is 2. So we understand that those spindle keep the maternal and paternal genome separated. So you start with maternal and paternal, and there will be 2 spindle, and when they come close together, those mechanisms keep them separated. So we not only understand, we not only identified that they are separated, but we also figured out why they are separated. And this is a recent manuscript. It's in bioarchive. So we also proved that our finding is valid in bovine or cow embryo. So it also has dual spindle formation, and that may keep maternal and paternal genome separated. So at some point, we will be able to prove that this is the case in human embryo. So I talked about the embryo, and now I will talk about the cell. So inside cell, we have lot of protein. So what we try to understand here is a fundamental process, which is cell division. So there, one cell divide into 2. You can see here, it goes through a lot of morphological transformation and transfer its genetic information to its daughter cell. So this is a fundamental process, but people didn't know what are the factors that regulate that process. So our group, before I came to EMBL, actually, we identified that there are 572 genes that control or guide this cell division process. But in later, we try to identify how they are related. So the problem is there, if you look at the protein is in different color, and the DNA is gray or white. So you can see the cell region. So there are around 572 protein like that, and we cannot see it in a single cell. So the problem is, with the microscopy technique, we could see only 1 or 2 protein together, this color, different type of protein. And when you image them, they will be in different orientations. So you can see that one is like this, the other one is, the orientation could be random. So you need to integrate them to understand that those proteins are in a single cell. So something like our objective is like that. We have one cell and we have all those protein together. You can see these dots here. And this green part you can also see here, and also the red component and blue component. So our target is to map all those experiments where we have this single cell into a single coordinate system. So what we cannot achieve with microscopy and experiment, we need to do it computationally. And this is really challenging because cell goes through different morphological transformation. You can see that they are quite different, and it also goes from one cell to 2 cell. So the way we deal with this problem, that we have different cells after segmentation, and we use these landmarks, which is DNA and chromosomal membrane shape. And those shape are used to generate a model, kind of an average model. And then this model is used as a reference to align or register, especially all those proteins, including the landmark. And then if you can do it, then you can integrate 3 proteins which were taken in using different experiments. So So it's kind of a virtual or canonical model where you can integrate all the protein together. So in microscopy, you cannot have it, but using computational method, you can do it. But it is also, we need to do it for different stages. So the problem is we can do registration, but we also need to deal with the progression of mitosis, means like baby growths are not same. So one, if you consider this is baby, and if it is month, like this is 30 months and 45 months, one baby has similar shape or size than the other at 44 and around 60. So we need to deal with this temporal alignment, means you can average them together, but you need to identify which are the similar size and shape that you can compare together. So so with this, we try to apply a method where we can align all these cells together so that the similar type of shape, they can come together. So this is called spatial alignment. So we need a temporal alignment, we need a spatial alignment, and we also need to automate microscopy. So existing microscope would not allow us to image what we wanted to do. So we were actually interested to cell division, but entire cell cycle is 24 hours, but our interest is only 1 hour. So we developed a macro that was coupled to microscope, so that tried to scan the image in low resolution, and before one cell goes to enter into mitosis, it activates high resolution imaging so that you don't need to expose the cell with phototoxicity before. So we can automatically generate high resolution images for the cell of interest. And we also do FCS technique where we can get the get the absolute number of protein so that we can compare data from different experiments. So after imaging, one of the important part is to segment the cell of interest. So you can see that there are DNA and also the cell shape. We need to segment the DNA automatically, and we also need to track because they can move a bit and also they will divide into two. So we need to detect automatically. There are two cells, so we call mitotic separation. So we do it in automatic way. So, and also we check whether we perform the segmentation quite well. We also detect many different parameters. So one is important, we need to align them. So we need to predict the cell division axis. So I use DNA shape to predict the cell division axis. So this is also a complicated process where you need to use spatial and temporal information to predict the cell division axis reliably. So we define cell division axis, then we need to define different standard stages. So after getting all these parameters, we can align different cells together to get a generic shape. And from there, we can automatically detect different transition points. And by doing that, we identified around 20 mitotic stages. So you can see that they are more precise definition of cell division. So we define them, discretize them in 20 mitotic stages. So the purpose of doing this is to annotate automatically all the cells and build a special model. So we take cells which belong to a particular standard time and we segment them. We define their predicted cell division axis, and we also determine the axis and use that to register them. So if you know this, you can align them. So after alignment, we also need to generate an average. So to generate average, I transformed Cartesian coordinate system to cylindrical coordinate system, so which is 3D version of polar coordinate system. So if you consider this is a surface point in a particular plane, so you can also represent this as a vector, which is for 360 degree, you will have 30 different radius. So something like that, you can represent this as a vector. So if you have many different plane, you will have many different polar coordinate system. You can integrate together to generate common cylindrical coordinate system. So now the way we perform this averaging, we have Cartesian coordinate system, we transform both cell surface, which is gray, and DNA surface, which is red. So we transform Cartesian to cylindrical and perform the averaging on cylindrical axis and perform the averaging. So if I repeat this for many different cells, I can generate the average cell from any number of input. So with this, we can generate average of all different mitotic stages, and their standard deviation means how different individual cells are with respect to the mean. Yeah. So now we have discrete model, and I can use those model to generate any intermediate shape. So we can generate continuous model from this discrete. So how to do it? So I transform them into cylindrical coordinate system and perform the averaging. So if you consider if I take stage 13 and stage 14, if I use 50% of these and 50% of these, you will probably realize from the distance that this is kind of middle between these two distances. So if you try to do it differently, that 75% of this and 25% of this, you will get this, and vice versa, you can generate different. So the cylindrical coordinate system allow us to generate from discrete to continuous model. So by doing that, we can also integrate all the protein into the model. So this is the average model, and these two cells belong to the same stages. So you can register and align and get protein distribution. So they are generated from multiple cells. And in the same process, we can integrate protein from different protein into the same cell. So our target is around 600 protein, although visualization would be very difficult. So this is just a snapshot of the model that you can integrate many different protein together, both spatially and temporarily. And we also can automatically classify different protein. The idea is that where they localize, based on that information, we can separate them into subcellular localization. Instead of saying protein name, we can say that those protein localizes on kinetochore or cell membrane or DNA or cytoplasm, something like that. So this is done by NTF. And we also can generate a lot of biological conclusion using this framework. So one example is this kinetics of chromatin-bound protein. So I'm not going to explain this, but just to mention that by doing that, we were able to show the community that this pipeline can be used to do a lot of things. And we got one collaboration request from University of Berkeley because they submitted a manuscript. Reviewer suggested that they should also check their method by collaborating us, because we have a different method that allowed to compare their data. So they contacted us, and we were able to match that together, and it was published in last year actually. So all the data we put in a website, which is mitocheck.org. We call Mitotic Cell Atlas, and this is the very first protein atlas for mitosis. So you can visualize all the proteins together. You can select them, which protein you want to see, and you can visualize. And this could be used by school students as well as the scientists who are doing research in system biology of mitosis. So you can see the results, you can quantify them, you can also look at localization data and source code and everything, raw data, everything is accessible from this website. So I think I will have 5 more minutes. So I will just touch another topic, which is nuclear pore complex. And this is a tiny structure which is between cell, so cell membrane. So cytoplasm is there and nucleoplasm is here. So there is a membrane, and this is a pore or hole between cytoplasm and nucleoplasm. So and this is really important to understand many diseases. So the structure as well as the assembly. So the problem is the structure is very small, which is 120 nanometer radius. So you cannot see the complete structure using confocal microscopy. So you need to have a bit better method. So super resolution microscopy arises for that. So what we did, just to illustrate how it works, you cannot see something because of blurring. So this is similar to if you see someone from quite far. So we don't see moon, the structure of moon a lot from the Earth. So the target is that the method that applies that you collect light, you switch off light, and you collect from individual molecule and try to detect that. So if you do it, you can get better picture of the same image. So you can now read it is GSDIM. So you can overcome the existing limitation of optics by doing this technique. And that's the reason 3 physicists got Nobel Prize in chemistry. So using this technique, we try to recover the shape or construct the shape of nuclear pore. So you can see that if you try to scan that, you can see a bit better structure than what we get from the confocal microscopy. So once we have this data, you can see these are the tiny structure, but you want to understand that. So if you just consider the previous topic where I integrated different cell or protein together, this is also similar problem. So we have a reference cell, which is protein, which is Alice. And then we also have SEH1, NAP107. Those are different proteins. So we want to see how those proteins especially locate together. So the way we did, we use the reference and we do perform averaging only the reference channel, and we get nice average. So if we use 8-fold symmetry as a constraint, we get nice average, and we use this channel as a reference to register the other channel, because this is the common element on all the protein. So we can average them. So by doing that, we could integrate all the protein together to generate an integrated 3D map. So you can see there are 5 different protein and also the reference Alice. So Alice is here, and the other protein you can especially resolve where they are. So in nucleoplasm or cytoplasm, or how they are. And we also can quantify the shape, that the radius are like this and their positions are like that. So we can get a lot of information. So diameter, distance between the ring, and also we can quantify their intensity, which is both axial and radial. So we also performed clustering there that they are not unique shape. So if you cluster individual particle within a particular protein, you can automatically find that they are different. So so what we did, we perform clustering and perform averaging individual cluster. We see that there are stages where you don't have lot of signal. So that relate to the assembly. So we call mini pore and mature pore. So this is also validated by one of our previous research that this when the NPC form, they initially work as a mini pore. They start in the nucleoplasm and then form it in the cytoplasm and become a complete pore. So using those three examples, I try to show that bioimage informatics, which is a part of bioinformatics, that can play an important role to study fundamental biological questions. So it is a different field from computational science, and my background, I was trained as a computer scientist. So I did my master's, bachelor, PhD in computer science, and then I was transformed to a applied scientist. So the point I want to make here that you don't need to be a biologist now to have a fundamental impact, crucial impact on the research in biology. So now all the research is becoming kind of multidisciplinary. You need to integrate expertise from different background. And by combining those together, you can do a lot of nice things and advance the state-of-the-art methodologies and also advance the knowledge which is available now. And since there will be also a lot of students here who has computational background, so I just want to say that you have enormous potential to contribute fundamental research in many different fields. So it doesn't need to be biology, it can be any other field. So that is the reason I just combine those three examples that you can have a deep impact in other topics. So this is the last slide that I want to acknowledge our collaborators, lab members, and yeah, a lot of people who are involved in this process. So thanks for listening. And I am now open to take any questions.
So the virtual floor is open for real questions. Expecting questions from the audience.
Should I stop sharing my screen or?
It's okay. It's okay. You can keep the sharing. I think the audience will make questions. I was wondering, Dr. Julius, really it's a mesmerizing presentation. When you were presenting many cells, proteins, their images, etc., I was thinking whether the background of Dr. Julius is from the biological science or from computer science. I'm really wondering how to concentrate on to the biological issues being a student of computer science and engineering. I don't have any technical question. I just want to request you, could you please highlight some fundamental research areas for our undergraduate or the graduate students and or young faculty members so that they can start with very fundamental problems related to these, because the results, the research that you have presented, I know it's highly sophisticated, requires very high-end device sometimes. So what is your suggestion for Bangladeshi scientists and researchers to start research related to this work? Thank you very much, Professor.
Yeah, thanks. And it's, I think, very useful question. So, and that's the purpose of all these meetings, I think. So one, I would separate it into two parts. So one is how to prepare ourselves. The other thing is what are the resources that we can access. So in terms of being prepared, so I think we are doing really great. We need background in mathematics or programming, and we need motivation and self-belief to start that we can do really great thing. And in terms of data, like now, I think we are moving to a research practice that when you publish something, you publish your raw data, you publish your methods, like source code and everything, so that other scientists or researchers, they don't have to mimic or duplicate the same thing. So it saves the energy and time. So one important thing would be that now if one wants to do research in biological science, so if you follow the manuscript, like in our group, we published whatever we published, we now publish the raw data. And yeah, so if I consider these three different projects, so we have the data. So if you consider the protein atlas, and now this machine learning, deep learning methodologies are being quite popular. So I perform segmentation there automatically. It was not deep learning on machine learning, but it does it automatically using sophisticated computational method. So this could be a base data where you have annotated result that was also obtained automatically, and you have raw data. Now, if someone wants to do deep learning or machine learning on the same data, so he will be able to access the data, he will be able to access the annotation, and then they can also play with the machine learning technique, whether they can exactly get the similar result. So those sort of thing could be a start to do or to progress. And in terms of biological knowledge, I think what you just commented before that, I don't know much about biology actually, but I know what I have to know. So it will come at some stage. But I think my time in Dublin City University, because my PhD was biology, pure computational science. I never worked with biological data. So when I was in Dublin, as I worked as a postdoc, so I was collaborating with biologists, physicists, but I was based in electronic engineering department. So I didn't have to know a lot about biology, but you get a bit of sense out of that. And then at EMBL, I could work with together with people in different backgrounds. So, and then you know a bit. So that will happen. So, but the most important thing is that you need to work with, you need to have your background and confidence ready. So that would be my suggestion for fellow students. Yeah.
Thank you very much. I just one supplement question to Professor Ajay,
Yeah.
with Professor Ajay. Is there any hardware requirement? I mean, yeah, knowledge part is okay. Is there any environmental requirement for pursuing this kind of research? Do you need any high volume computational machine or microscopes or something?
So to generate the data, it's you need like sophisticated machineries. Like it's an image. So what I get, but what you want to see inside the cell, this is a process, and it takes time to actually the technique is, if you want to see one protein, you need to identify in the genome where it locates. You need to target that, you need to mark the marker, and then prepare the cell line, and it goes to the microscope, and you need to do imaging. So I think that would be the part you cannot contribute from, like this is also funding, like one microscope, only microscope is €1,000,000. I think Professor Rajak, Professor Ahad, they also came, and they at least they saw confocal microscope here. So so I think that, and also daily research requirement, preparing material and consumable, that will be really expensive, and not in developed world, not all the university effort to do it. Like that's why people call EMBL is Mecca of biological science. They don't have to consider the money, like it's not a limitation. But once you have the data, then this data is now accessible. So as a scientist, you need this data, and you can work with that. So that will be the things. But one advantage I have now, because our data generation and computation analysis work in the same facility, and you can say who prepared the data, you can plan together, you can spend months to have the data in a way that allow you to perform the analysis in a better way, so that if you can solve some of the problem during imaging, you don't need to deal with those problems in the analysis. So this is an advantage that I have. But when you have a data given by a collaborator or from publicly available repository, you don't have that flexibility. So still, I think now it's really, if someone wants to work with data in a different field, they will be able to have it. Yeah. And so
much.
maybe another point, probably just computational requirement. So all the research data that I put here could be generated using a conventional machine, which is not so bad, but we also have like very sophisticated parallel processing and clusters, and as well as data center, because we generate terabytes of data, and that is good for computer science, that people now generate the data in a year, one scientist that probably 100 people cannot even see what is there. So you need automated quantification and QC to deal with those data. Yeah. So that, but this is not a bottleneck, that you need to have sophisticated parallel processor and yeah.
Thank you very much. I think Professor Atik Ahad raised his hand. Yeah, please.
Okay. Yeah. So thank you. So would you please go back to the slide of Joe Karim, where you mentioned about the 4D clustering?
Yeah.
Yeah. So yeah. So just, I mean, here the clustering is done in so minute level. Definitely, you didn't consider the traditional image clustering method. So what kind of strategies you considered, or what are the things we need to look into for this kind of cases, the clustering and so on? Any point on that? This is the question number one. Number two is that, I mean, you said that EMBL is the Makkah of money. What about the salary?
And not money, but facility. Because all these European countries, they put money here so that you can do whatever, you can buy whatever you want, and you can do research here. So that's why people call it Makkah. Yeah. And what's the question? Sorry, I interrupted you.
Okay, the first question is the clustering. How did
Yeah,
you?
first question I understand, and second one.
A second one is was a joke. So how much you get from those
Oh, okay.
lots of money?
Yeah. So first one, actually, this is I regretted that I could not explain, but you gave me the opportunity to explain. Now this has been great. So you see interest point cluster here. So there are 80 different features, and this is also not deep learning. So after segmentation, so you see that there are different proteins. And when we have this, we try to extract different feature from there. So one of the feature, many features are also taken from SARF. Yeah. So, and also the position and the amount of protein, all those information are put together to generate this 100 size vector. So there are 100 features. And I mentioned that we have 100 feature for each of the cell, and then we have 20 different mitotic stages. So which we identified by temporal warping, so time warping. And there is another dimension, which is we have 600 protein. So 100 features, 600 protein, and 20 different mitotic stages. So that defines our research. So although we didn't have 100 because we imaged, like imaging one, preparing experiment, it takes a couple of months. So it's a experimental limitation. So we had around 50 cells. So 50 different protein, 100 cluster, and 20 different mitotic stages. So our purpose was to have, instead of protein, we wanted to have that this protein localizes on something, so that now you can see this Aurora KB is a protein, and a biologist should understand where it localizes. So you see that initially they localizes on DNA, so which is not completely cell, and in the middle it's also DNA, and then it goes in the middle, we call mid-body, and there. So for other protein, you can also see that at each time point where those protein localizes, and at which amount. This is amount 0% to 100%, and this color means that in mid plane, that particular protein PLK1 localizes on those part at this percentage. So he used NTF, which is unsupervised, that is soft clustering, where you just reduce the dimension. Instead of protein cluster, which is 100, size is 100, you get all the protein. You also keep the standard mitotic stages, and then instead of feature, you now say it's protein localization. Then protein localization, we know, is centromere or chromatin or spindle centrosome. So these are the kind of position. So chromatin is the DNA, midplane is in the middle between two centrosome, cytoplasm is just in the middle of this land, and centrosome is, we don't have centrosome here, so but you saw that there are two different dots. If I draw something like this, there will be two different dots. Where can I, maybe something like that. So if you, this is a cell and this is DNA. So centrosome would be something like this, these two dots, which generate spindle and try to control those things together. So instead of feature, because feature doesn't tell us anything, intensity and size, shape, but you can say that this protein localizes us on those subcellular structure, and and then you also can quantify. So the last point I want to make, that why this is important, because the protein that localizes on the same subcellular structure, let's say kinetochore, they are friends. So let's say for one disease, 20 different, one protein is related. Let's say for a particular disease, this protein, we know that a particular protein is related. Now, if you know that other protein, other 19 different protein also has similar localization, they also localize same, they stay in the same place, if you want to discover drug, you need to also look at what are the role those 19 protein are playing. So something like that, if you want to steal something, and then if there is one guard who is just taking care of the things, it's not enough. If there are other players in the same place, so you need to consider those. So if you try to mute or inhibit those protein to deal with the disease, you probably need to deal with those other proteins. They have similar localization. So that's the biological understanding that we want to have, that those protein localizes in the same subcellular structure. Probably they have some relationship at those stages, and then to discover drug as well as to understand the behavior of a disease, you probably need to understand the function and role of those other protein too. So yeah.
Thank you
Right.
so much. Yeah, thank you, Julius.
Sure. Thank you so much. So I think that's done. Thank you very much, Dr. Julius, for being with us. And I'd like to hand over our microphone to Professor Abjad.
Thank you very much, Professor Dr. Mamunur Rashid, for chairing this session. I'm really impressed to see the scholarly discussion we had at the very last keynote session here in this STI 2020. And I always see lively all around the whole sessions, keynote sessions, technical sessions, especially the presence of Professor Dr. Atikur Rahman Ahad was always highly vibrant. And I really appreciate the keynote speaker for his valuable speech. Dr. Julius, could you please close your sharing so that our rapporteur can share the appreciation certificate? Yeah. May I now request the respected chair of this session, Professor Dr. Mamunur Rashid, to confer the certificate of contribution to Dr. M. Julius Hossain.
All right, it's virtual. Presented to Dr. Julius for his contribution to STI. Thank you.
Thank you very much.
Thanks a lot for this, and
yeah.
I appreciate.
Thank you. Thank you very much.
Thanks for the invitation as well again.
Always your presence is encouraging for us. And STI 2020 organizing committee is also immensely happy to confer the certificate of appreciation to Professor Dr. Muhammad Mamunur Rashid for his presence in the campus. In fact, Professor Rashid and myself is sitting in the same desk opposite using two computers. So we are here. So thank you very much, Professor Mamun, Dr. Shariful Islam, Atik Ahad, and many other distinguished guests here. We are about, it's 5:30. After 1 hour from now, we are going to start our award giving ceremony. We would like to invite you all there, and the award given ceremony will be chaired by the Honorable Vice Chancellor of the University of Bangladesh, Professor Dr. Ghulam Samdani Fakir, and where we will have amongst us the Honorable State Minister for ICT, Government of the People's Republic of Bangladesh, Mr. Junaid Ahmed Palok, as the chief guest. So thank you very much, and I'm inviting you all again to that session. And thank you, Rapporteur Sultanum Habiba and Mahmoud Rahman, for your nice supports for coordinating this session. Thank you very much. Assalamu alaikum and very good night to you all.